Okay, good morning everyone. Gives me great pleasure to introduce to you this morning Jason Waithman. He's from Telethon Institute. He's a molecular and cellular immunologist and obtained his PhD in 2008. He received his training at University of Melbourne, Walter and Eliza Hall and the Ludwig Institute for Cancer Research, so all the top places in Australia. And has returned to Perth in 2012 and has established his own group over there on tumor immunology, and he's going to tell us about his work as well. Thanks, Jason.
So good morning, thanks for the invitation. So I'm going to tell you two stories today, two different projects that two different PhD students have sort of led. So the first one is the resistance to adoptive cell therapy by silencing of immunogenic antigens as a result of complex epigenetic events. Okay, so just to give you a bit of background, cancer therapies, surgery, radiation and chemotherapy have long been sort of the standard treatments against cancer, but recent clinical trials have really demonstrated that immunotherapy is quickly becoming a pillar of treatment for some cancers. I mean, even the skeptics now can't refute that, as people are actually being cured of their disease. So one of these therapies is called adoptive cell therapy, and the first generation sort of came out of the NIH with Steve Rosenberg. So basically what this involved was, especially in melanoma patients, where you can harvest, originally it was tumor infiltrating lymphocytes, and they could get them out from the blood, and find the tumor specificity, sort of grow them up ex vivo in a huge vat and make a huge army of these, and then sort of put them back into the host and have them sort of hunt down the cancer anywhere it is and eliminate it. We've now got second generation and third generation therapies, cellular therapies like this coming online, where we can genetically engineer T cells, such as the chimeric antigen receptor T cells, where you can put in a CD19 specificity to B cell malignancies such as lymphoma, leukemia, etc. So as I mentioned, adoptive cell therapy can be very effective in some patients, resulting in remarkable cures. But there's also the other side, where we see the development of acquired resistance by the tumor cells, and it remains a real barrier to effective therapy. And this is probably best illustrated by the CAR therapy, showing that in both pediatric and adult populations, that the cancer can sort of respond by either downregulating its expression of CD19 through loss of chromosomes or even alternate splicing events. So we've been working on a model of adoptive cell therapy that's sort of like the first generation one, where it's sort of trying to mimic a tumor infiltrating lymphocyte therapy, but we use transgenic models. What we've got here is two solid cancer lines, the murine-derived B16 melanoma cell line that we've made to express a model antigen ovalbumin, or OVA, and AB1 mesothelioma expressing the influenza HA antigen, so AB1HA. So both well-characterized model antigens. And we have access to the OT1 TCR transgenic and the CL4 TCR transgenic animals. And what these animals have been engineered to, that all of their CD8 cytotoxic T cells have specificity for these model antigens. So the OT1s have OVA-specific CD8 T cells, and the CL4s have HA-specific CD8 T cells. So what we do is we activate these T cells just like you would in any clinical adoptive cell therapy protocol, by peptide pulsing splenocytes and using LPS, and basically expand them for 4 days in IL-2 containing media, so we get a huge amount of effector cells. So just like the clinical scenario, prior to adoptive transfer, the mice are given a lymphodepleting regime that sort of increases, it's known to increase the efficacy of the transferred T cells. So you can do it by chemotherapy or radiation. So we use radiation, and expose the mice to about 550 rads of total body radiation. So it's a non-myeloablative event for them. So then mice receive adoptive transfer of 10 to the 7 activated cytotoxic T lymphocytes once tumors are well established. So either day 8 or day 10, depending on the model. And then we monitor tumor growth over time. And what we can see, the results from these experiments, is that if you just look over here, and this is the B16 OVA plus the OT1 system, we can see that mice that just received TBI, we get exponential growth of tumors. But those that receive the adoptive cell therapy, the tumor-specific CD8 T cells, we can see a sort of regression of disease, some stability here, some sort of equilibrium event, and then we see rapid outgrowth of tumors in all mice. We can't cure them in this system. And very similar results in the mesothelioma treated group as well, that you can see the kinetics of the not treated versus the treated group. So these results demonstrate that there's some sort of mechanisms of tumor resistance to this adoptive cell therapy. And as I mentioned before, these tumors can develop a range of acquired resistance, such as suppression of immune effector cells, loss of target antigens, just to name a few. So we had a closer look at the T cells infiltrating the tumors, and as well as those circulating in the animals by looking in the spleen. And we just started to phenotype these cells just for basic checkpoint markers, such as program death 1 or CTLA4, very well known sort of exhaustion models or checkpoints that sort of suppress CD8 T cell activity. And what you can see is that PD-1 is upregulated in the tumor in both model systems, whereas it's not in the spleen. So the tumor microenvironment is sort of impeding probably the ability of these T cells to really do their job. There was a trend that CTLA4 was upregulated in the tumors, although it wasn't significant. And interestingly, in the B16 ova model, there was a loss of effector function as measured by the ability of these T cells to express interferon gamma and TNF alpha as compared to their circulating counterparts. This wasn't observed though in the mesothelioma model. So it looked like we're having some sort of suppression event that's pretty well characterized. So the next experiment that we did was we said, okay, if these T cells are starting to get sort of exhausted and running out of steam, why don't we just put in fresh activated T cells once the tumors start to come out of this equilibrium phase and start to relapse. And so, as you can see here, the results from this experiment is when we put in the second round of adoptive cell therapy, we didn't even put a dent in the tumor at all. So the tumors just continued to outgrow at an exponential rate. So this got us now looking more closely at the tumors, and basically in the model that we've set up, and the tumor cell lines that we've engineered co-express markers as well as the model antigen. So for example, in the B16 ova model, the ova antigen is controlled under the same promoter as green fluorescent protein. So we can measure overexpression by GFP as a surrogate marker. And in the AB1HA model is linked to antibiotic resistance. So when we took tumors from relapsed animals and basically grew them ex vivo, we could perform flow cytometry to just look at the expression of GFP in the B16 ova model. So if you could just look here, you can see that these are the original cells that we put in where we didn't perform any adoptive cell therapy. So these are explants from mice that just didn't receive treatment, and you can see nice expression of GFP. However, those that received adoptive cell therapy or immune pressure, we lost expression of GFP. So similar to the background levels of an untransduced B16 cell line. In the AB1HA model, when we started to expose explanted tumor cells that were tumor-derived explants to the antibiotic, we could see that basically they all started to die, those that had received therapy. So again, a loss. That is a surrogate marker that we could see a bit of a loss of the antigen expression in these cell lines. Okay, so we know that tumors are genetically unstable. They can lose parts or even whole chromosomes through karyotypic loss and aneuploidy. And our immunogenic antigens lost through these mechanisms. So we had a bit of a closer look at the tumor cells. So we just performed PCR on the genomic DNA of the B16 ova explants, and we could still see a fair amount of definitely less in some cell lines, but this is just three tumor explants that we looked at. This one here, you can see there's still a whole lot of the ova construct or the engineered ova antigen in the genomic DNA, meaning that they haven't kicked it out like a chromosome or something like that. And similar results, over here we're looking then at the RT-PCR, so we're looking now at the mRNA level, the expression of these antigens. And you can see that it's pretty much lost in these cell lines. So it seems to be some sort of what we think is an epigenetic event. Okay, so epigenetics in cancer. Cancer cells often display localized hypermethylation around specific gene promoters, altering gene expression. So this is mediated by enzymes such as DNA methyltransferase. So what we did is we started to treat these cell lines with azacitidine or decitabine to see if it could restore the antigen expression. So if we take off those regulation markers, can we now start to see GFP upregulated? And what we could see here from the data is you can see that we see some upregulation, like in a minority of the cell populations, we can re-express over. So it looks like that this mechanism is occurring as part of the escape mechanism by the tumors under immune pressure. So what we did is we basically, after this treatment, we started to sort the cells. And we sorted them into the refractory population and the susceptible population as measured by GFP expression. And from this susceptible population, what we then next did is actually we sorted single cell populations of these and let them grow up. And so we've got many and many generations of cells here. And then we just faxed them again for GFP. And we were quite astonished that actually the GFP expression was quite stable. So like once we take off these repressive marks, they can carry this this information through multiple generations. So it's sort of a positive result in the sense that if this was to work in the clinical setting, you wouldn't have it quickly shut off once you take them off treatment. Okay, so what about the refractory population? So there's a lot of different ways, a lot of different epigenetic events that can occur. So in collaboration with Mark Cruikshank, who runs a group at the Telethon Kids Institute, and we started to investigate a lot of these different mechanisms. He has a collaboration with GSK and has a whole library of compounds that target different epigenetic events. So this just gives you a whole list of the drugs that we used in these experiments, where basically we had the refractory population and we basically exposed them to single agents with the hope that we could turn on GFP expression or over the antigen. And unfortunately, we didn't get a single hit, which we were a little bit surprised about. And this has gotten us to sort of to the conclusion that this is probably a very complex event. And at this stage, the next series of experiments that we're doing to finish this off is we're basically using a chip assay where we can basically use antibodies against some of these repressive events and start to try to pull down the histones and the genomic DNA and do a PCR across to see if these mechanisms are involved, and to try to get an idea if we need, what kind of combined therapy would we go in with. It's sort of a, instead of just shooting in the dark, I guess. So in this refractory population, as you can see, we first had to check whether or not that the OVA gene was still in the refractory population. Again, there's just a PCR on the genomic DNA, and as you can see, here's just the control cell line, and basically just a titration of cell numbers down here, and this is sort of the limit of detection. And as you can see here, this explant, at least from adoptive cell 1 of the AZA or decitabine treated, it basically looks the same. So it seems like the majority of the escape mechanism, at least in this certain explant, is most likely an epigenetic event. But again, we did see differences across the 3 cell lines that we looked at, where we see a varying degree of this event occurring, meaning that aneuploidy or karyotypic loss, which has been described previously in the literature, is also probably occurring as an escape mechanism. Okay, so this is what we think is happening, so that as the cancer is getting really hammered and under a lot of immune pressure, basically it's has the ability to down-regulate the target antigen by, we don't know, agent X, Y, Z, where we're going to try to find out. And basically, you know, the positive note of, I guess, the study is that the re-expression of the immunogenic antigen is highly stable. This is with the DNMTs. But as I just said, multiple epigenetic events drive acquired resistance to immunity and tumor heterogeneity. And I think this is really sort of an important study because right now a lot of the clinical trials that are going ahead are really just going in blind with single agents. You know, panobinostat, I know, is just going into a phase 1 clinical trial for kids with brain tumors with very little data to support any effect. And we're sort of hoping that this study will sort of inform on that approach, at least to show that how complex this might be and that more research needs to be done. Okay, so this work is done predominantly by a PhD student, as I mentioned, Ben Wiley in the lab in collaboration with Mark Cruikshank's group, and all the mesothelioma work was done in collaboration with John Chi, a postdoc in the tumor immunology group at the University of Western Australia, and just it's been supported by, the NHMRC and the Cancer Council. Okay, so I'm going to tell you about the second story. Sorry, I tend to talk pretty fast, so I'm sort of whizzing through this. Okay, so this one's all about interferon alpha subtypes and their ability to drive distinct anti-cancer activities. Okay, so interferon alpha subtypes. It's been long known. Interferon alpha has been studied probably for 50, 60 years, and it was first discovered as its potency against viruses, and then later its role in cancer has been described. Now, multiple subtypes of interferon alpha exist, and that's well acknowledged in the field, and sort of every introductory paragraph of a paper sort of starts out this way, and then in general, the majority of them go down the line, we used one of them, this is how they all behave. So we sort of had a question on, do the individual interferon alpha subtypes, can they drive distinct anti-cancer activities? Why would you have 12 different interferon subtypes? Did they serve a purpose? Why does the body produce so many? That's our basic question. Okay, so the methodology that we sort of employed to investigate this was to basically engineer the B16 melanoma cell line with individual interferon alpha subtypes. So these cell lines secrete, for example here, interferon alpha 2, 4, 5, 6, and 9. And basically we confirmed this active secretion by a bioassay. It didn't appear that they upregulated class 1. And really the proliferation, at least in vitro, was sort of similar across the cell line. So we did a bit of characterization prior to implanting them into just wild-type black 6 mice. Okay, so here's the results. So if you transplant just the empty vector control, so just B16 melanoma cells expressing GFP, you get rapid outgrowth of tumors in 2 to 3 weeks. So very large masses form rapidly. And all the different interferon subtypes had, to some varying degree, basically inhibited tumor growth and progression. But the first results that came out from this experiment that were quite interesting and sort of took us down a certain pathway that I'll talk to you a little bit about is, we noticed that some mice didn't develop tumors in some of the cell lines, so they never developed any masses, any palpable tumors. Okay, so these two are B16 interferon alpha 2 and B16 interferon alpha 9, and we have interferon receptor knockout mice. So all of these interferon alpha subtypes signal through a common receptor, and basically when we put them into these IFNA mice, we can basically see that we now lost the effect completely. So the only cell in the system that can actually respond to the interferon is the tumor cell line itself. Okay, so it's sort of led us down the track that the immune system's most likely involved here. So we also have RAG knockout mice. So the RAG knockout mice lack T and B cells, so the adaptive immune system. And basically, when we put them into the RAG knockout mice, we completely lost the effect. All the mice develop tumors, so we don't see any of that protection that was observed previously. We then asked if it's something to do with the cell line, so we set up an experiment where we took these B16 interferon expressing cell lines, and prior to implantation into a black 6 mouse, we mixed them just with wild type cherry cells, okay, to see if now we could see rejection of the wild type cherry cells as well. And basically, here's the results we did. As compared to obviously the cherry cells implanted on their own, they all formed large tumors, as I said, in about 3 weeks. So we could see the effect transferred to wild-type bystander cells. What we next did was we engineered these cells to express model antigen, herpes simplex virus derived glycoprotein B, and there's good reason for that. We have TCR transgenics to those to that protein, and basically also put in firefly luciferase so that we could do longitudinal imaging if we wanted to. So, and then again, we planted those into black 6 mouse, and now remarkably, and this is probably needs to be updated, I think it's the last data I saw was about 84%. We saw a huge increase in protection once we put these model antigens in and sort of allow the immune system to better be able to see the disease. So we saw a huge increase in protection as compared to this cell line in, again, the IIID mice. They all developed tumors. Okay, so the luciferase enables us to do imaging of these animals, so it's a non-invasive procedure that we can image over time as the substrate basically glows, and that's what you're seeing in the tumors. So you can just see here in the interferon alpha receptor mice, the animals that we've imaged, you can see that the tumor masses just grow uncontrollably, and you can sort of measure tumor size as a surrogate marker by the amount of photons per second of illumination that you see from the luciferase. So over here in the black 6 animals, what we see in a lot of animals is basically we can start to see tumors starting to form, and in general around day 14, 17 to 20, most animals have been able to eliminate their cancer, those that never develop a palpable mass. So tells us that these interferons are very, very good at modulating sort of the adaptive immune system to really help control nascent disease, if you like. But then very interestingly, and again, I need to probably update this figure, we've actually got some mice that we can see this event out to day 250. We see some animals that enter into sort of a stage of equilibrium, if you like. So they never get rid of their disease. We can just see the disease sitting there, sort of, like I said, up to 250 days. We see a signal, sort of suggesting that there's two pathways or two sort of immunological events occurring, either elimination or immune-mediated equilibrium. This has been a pretty well-described phenomenon with the immunoediting hypothesis, sort of that Rob Schreiber has published previously. So we're quite interested in this event because it hasn't, the mechanisms associated with it haven't really been well characterized. Okay, just to further characterize like what's happening in our system. So we asked a very simple question: is interferon alpha 9, does it act locally or does it systemically? So the sort of experiment that we did here is we have tumor cells that have been engineered to express the glycoprotein B, so they don't have any interferon, so just B16G B cells, no interferon in the system. We take that and we plant it, implant it on this side of a mouse, and also in another mouse on the same side here. But then on this mouse, we also implant our B16 interferon alpha 9 GB luciferase expressing cell line to say, can the immunity that's sort of generated to this tumor and modulated by the interferon, can it now protect against a tumor on the other side? And what we see is that it doesn't, basically. So it really tells, this experiment tells us that this is all happening in the local microenvironment. So which adaptive cell is involved in this enhanced immune protection? We started to do experiments where we, prior to tumor challenge, we would deplete cohorts of mice with either, deplete them of CD4 cells, CD8 cells, or CD4 and CD8 cells together, as well as a no depletion group, and then transplanted our alpha 9 B16 expressing tumor on. And basically what we can see is that we basically see in these experiments 100% protection when we don't deplete. A subset of the cells that when you deplete CD4 still remain tumor free, so suggesting that CD4s, while they might play some sort of role, they don't play a major role. But we completely lose the effect when we deplete CD8 T cells. This is a CD8 T cell driven event. Okay, so we have GBT1 TCR transgenic mice in our colony, and basically these are similar to the OT1 and CL4 system that I previously described. These basically are a source of CD8 T cells that are all specific to the herpes simplex virus derived glycoprotein B. So I'm going to get into some pretty hardcore, complex immunological experiments of what we're thinking of doing next. So basically what we do, these are IIID mice where we know that these B16 interferon alpha 9 lines grow very well in. We basically implant various cohorts into these IIID mice, and basically we also transfer naive GBT1s into one of the cohorts. So now these naive GBT1s are the only immune cell population that is responsive to the interferon being secreted. So these have an interferon receptor on them, the transferred cells. And then over here, we basically transferred pre-activated T cells, so effector T cells, if you like. So the GBT1s, and as a control, we have crossed them to the IFNA background. So because the affected GB-specific cells that I showed you previously, they can actually do some damage to the tumor and cause it to regress a little bit. So it was appropriate to have the correct control. And these experiments are basically asking, is there a direct effect of the interferon alpha-9 on the T cells? And what we sort of found is that there isn't, so it's a bit more complicated than that. So I'll just walk you through the data. So when we don't transfer, here's what the kinetics of tumor growth look like in the interferon receptor knockout mice. The naive GPT-1s, basically we saw no increased survival, so the interferon's not directly acting on CD8 T cells as they differentiate and enter the tumor microenvironment. And the effect is, as you can see, there is a bit of a survival advantage over the no transfer group, but that's because they can recognize and do some cytolytic activity, etc. But it's no better than when we transferred in the GBT1 interferon receptor knockout control cells. So if anything, the control did better. We saw a mouse or two that actually did survive. Okay, so what we're doing next is basically using what I call a subtractive in vivo approach to sort of try to tease out what other parts of the immune system are involved in this process, so that we know where this type 1 interferon subtype is where the modulation is occurring. So the first experiment that we're setting up is just a basic reverse chimera where we basically replace the bone marrow of a black 6 animal with the interferon receptor knockout bone marrow. We wait 8 weeks for the immune compartment to reconstitute. So you now have a scenario where the parenchyma or the stroma is all sort of derived from the B6 animals, so they're responsive to the interferon secretion, yet the whole immune compartment, so the hematopoietic derived compartment, is refractory. And then the reverse chimera is just the opposite of that, so B6 into IFNA knockout. So basically, when we now transplant these alpha-9 secreting cell lines into these chimeras, we can determine whether the interferon alpha-9 is acting on the parenchyma or the hematopoietic compartment. And obviously my gut feeling is it's on the hematopoietic cells, but we have to do the simple chimera first to sort of ensure that. With that sort of hypothesis, we're looking in further to do some mixed bone marrow chimeras. So here what we do is we irradiate an interferon receptor knockout mice, and we basically reconstitute it with bone marrow from 2 different animals. So in this animal, a RAG knockout, and this one, an interferon receptor knockout. And what that means is as the immune system kind of resets and develops after those 8 weeks, you're going to get innate cells. So your whole innate compartment can still see the interferon. Yet this animal is important because now the whole T cell compartment will reconstitute, B cell compartment will normally reconstitute from this bone marrow, but none of it can see the interferon. So it sets up a basic experimental animal system where we can tease out our innate immune cells involved. Okay. So as another mixed bone marrow chimera that we've just got the RAG gamma chain knockouts in from the Peter Institute. This is very similar to the RAG knockout in the sense that it doesn't have any adaptive immune cells, but the common gamma chain knockout means that all innate lymphocytes are gone. So now it just leaves cells of innate myeloid origin are still in the system and can still see the interferon. So that would be things like dendritic cells, macrophages, etc. So basically, again, our readout is tumor growth. So I think that these experiments will be very informative to sort of determine which cell population is involved in sort of assisting the CD8 T cells to go and do what we want them to do, and that is destroy the cancer. So yeah, this is a bit of a work in progress. And the results from this experiment will also inform us on to which way to go with flow cytometry experiments to start to try to investigate a little bit further. What are we seeing? We're seeing if we see an effect in these animals, is something happening with the dendritic cell compartment, and we've got some animal systems where we're pretty well set up to sort of monitor the cross-presenting dendritic cells, etc. Okay. So the other thing that we're doing right now is we know that at the pointy end it is the CD8 T cells, and our model system allows us to put in our GBT1 cells because the B16 interferon alpha-9 secreting cell line also express the model target antigen. And we can now start to sort of look in the effector site, the draining lymph nodes, the spleen, etc., to see if the T cells look different to when there's no interferon alpha 9 in the system. So we're going to look at differentiation markers such as CD103, CD69, sort of something that we're sort of thinking of that might be involved in. This is from another project with a collaborator in Melbourne, that we've sort of got some data showing that tissue-resident memory T cells are very good at, not really good at killing tumors, they're really good at driving it into equilibrium. And as you can see, we had sort of that equilibrium event from the imaging data. We're going to look at, is there a difference in effector molecules, granzymes, CD107A, so things involved in basically cytolytic activity, as well as, your basics, interferon gamma, TNF alpha, so the secretion molecules that might impede tumor growth. And lastly is looking at the inhibitory receptors, so the checkpoint inhibitors, so that we know that sort of dampen down immunity, post-viral infection or in the cancer setting. So, and these are the markers that in collaboration with Vanessa Fear at UWA that we're going to look at. Okay, so I've told you a fair bit about interferon alpha 2 and 9 and how we think that they might be sort of polarizing the immune system and modulating them to have an enhanced tumor effect. And we've sort of focused on them more than the 4, 5, and 6, but we've got some very interesting results on the 4, 5, 6, and we've started to get a little bit more funding, and we can sort of push on and sort of try to identify what's happening there. So this is the results again that I showed you previously when I started this story of the alpha 4, 5, 6. So tumors develop in all mice, but can be very, very slow. Such as here, if you look at alpha 4, we've got mice going out to 150 days, and still tumors haven't reached maximal size. They for some reason they tend to get really itchy, and the tumors are really sort of kind of they've sort of imploded, and then it tries to grow out in another area, and then it gets kind of clamped down on, and the scabs don't heal overtly well. Like the disease and the immune system are really having a pretty full-on battle, if you like. When we put them in the interferon receptor knockout mice, so again now the immune system can't see these interferons, we completely lose all the effect. Okay, interestingly, when we put them into RAG knockout mice, we still see quite a significant amount of protection, at least at the local tumor site of implantation. So that was a bit of a surprise to us, in the sense that it looks like these guys, at least for the local tumors, the innate compartment is being modulated to provide that protection. But all of these mice developed a very severe sort of respiratory condition, and they had to be culled for animal welfare reasons. And when we did a closer examination, and sorry for the sort of nasty picture, but they all had widespread metastases. So the reason why they're having trouble breathing is that the disease had spread to the cervical lymph nodes, and it was sort of clamping down on their windpipe, if you like. And you don't usually see metastases in this model, so this was quite a unique event. And basically what we did is we started to quantitate that. So what we did is we compared RAG knockout mice and black 6 mice, sort of side by side, implanted with these cell lines. And when the RAG mice died, whatever its tumor size was, we let the B6 get to that same size, if you like. So the timing's different. In fact, it's in sort of favor of it's working against the black 6 mice because they tend to live a little bit longer. So it takes a little bit longer for those tumors to get up to that size. And then we just basically quantitated the presence of metastatic disease throughout the lymphatic compartment and the lungs. And basically what you can see is in the RAG mice, this is just measuring the tumor size, for example, a lot of lymph nodes involved. Whereas in the B6 mice, we could see the presence of metastases, yet it wasn't widespread. Sorry, it wasn't very large. It definitely didn't get that sort of severe respiratory phenotype, almost like it was controlled by the immune system. And again, the same sort of incidence of metastases to another organ, such as the lungs, was present in the RAG knockout mice, and I don't think we've ever picked it up in the black 6 mice. So the interpretation of these results is these type 1 interferon alpha subtypes, they enhance the innate immune control of the local tumors, but there's some form of adaptive immune control of the metastases. Okay, so as I mentioned, we got the rag gamma chain knockout mice in from Melbourne, and then we're just expanding that as part of our colony. And we're gonna, the next experiments that, at least from the local effect, we're gonna start to transplant these B16 alpha 4, 5, and 6 tumor cell lines into these animals. And what we're really sort of interested in seeing is what if there's a split in the protection in these animals, like that you know, this basically will tell us if innate lymphocytes are being modulated by these interferons. So for example, let's say that tumors are controlled in this one and this one, but not that one. It would sort of give you an indication that there's also a role for innate myeloid cells potentially being modulated by a specific type 1 interferon. So we're quite excited about the results from this experiment. And again, it will also help inform us on flow cytometry, etc. So as far as the metastases go and the role of the adaptive immune system in this, we've started to set up a series of experiments where we just start to deplete different populations of T cells, sort of like what we did in the alpha 9 experiments, to basically ask, like, okay, so which part of the adaptive immune system is involved in providing this protections against metastatic disease? Something that we're quite interested in doing is also do the imaging here. So one thing that we don't know is that protection that the adaptive immune system is providing, is that actually at the local site? So do we see an early metastatic, or sorry, a late metastatic event? Or do we see an early metastatic event to the draining lymph node, for example, and then the immune system is clamping down on it in there and limiting its ability to spread further down to the chain of the lymphatics, for example? So we just started to do some imaging experiments to address that question. And as well, once we sort of have a bit more information on that, this enables us to phenotype the T cell population and see exactly what's going on there, how are they doing this. Okay, so in summary, I hope I've showed you today that with this story that the interferon alpha subtypes can drive distinct anti-cancer activities. So I think that we've addressed our initial question and hypothesis. And that the interferon alpha 2 and 9 provide CD8 T cell immunity with the ability to completely control nascent tumors. And interferon alpha 4, 5, and 6 enhance innate immune control of local tumors, but also adaptive control of metastases. Okay, and this project is being led by a PhD student in the lab, Anthony Bazai, with the help of a research assistant, Tegan Wagner, and Bree Foley's postdoc in the lab, and just acknowledge all the core facilities in all our studies, and again in collaboration with Vanessa Fear and Thomas Gebhardt at the University of Melbourne. Thank you for your attention.
Thanks. Jason, any questions?
Jason, I was just wondering, with the different interferon alpha subtypes, is there much known about the relevance of the different expression levels, and how do you ensure that that mimics the natural sort of levels of expression of those different cells?
Sure. I mean, I guess what we're thinking is that to translate this, I think, is where you're going with this, and if it's very difficult to address what you're asking me, because there aren't very good reagents necessarily to quantitate that. They're starting to see a little bit in the literature of, at least from an RNA perspective, of at least different subtype expression when certain things are happening. But yes, very difficult to quantitate that, and dose is something that we've obviously been thinking of, but it's very hard to address, because this could all just be a dose issue. I totally agree with you, but you also don't know if, you know, as this interferon alpha is being secreted or other interferon alphas being turned on. There's a monoclonal antibody available, but just recently talked to the group that, you know, Rob Schreib's group, that where it was generated, it only cross-reacts with maybe half the subtypes. So, very good question. What we're thinking is more from a clinical perspective, is to wrap this up into like a cellular sort of therapy, if you like. So when immune cells go into the tumor microenvironment, then they can turn on interferon when they engage the tumor, or if they're part of the stroma, etc. So that's how we're thinking of, let's just drive it, let's just make it happen.
More questions? Francois? No, Siobhan. I always do this.
Another question about the interferon alpha subtypes. Is it known whether the different subtypes bind the interferon alpha receptor with different affinity?
Yes.
They do?
Yep.
And about the interferon alpha receptor type 1 and 2?
I think there is some indication in flu systems that you can get interferon signaling through the homodimer receptor 1, but I'm not sure if that's been seen in other systems, and I haven't looked into it much further, but basically most of them signal through the heterodimer.
So do you have a correlation between binding affinity and effect you can see in either T cells or innate cells?
We don't know the targets yet. As you can see, we know it's not the T cells directly. So I'm more of a cellular immunologist. I do dabble in the molecular side of things, as you can see by making the models. But like as far as all the signaling pathways and all that goes, as soon as I hear that stuff, I'm lost. You know, I just went to an interferon conference in Canada, International Keystone Conference, and it was all about the intracellular pathways, and it's a huge area that I'm just not across, so I can't address what you're asking. So I think there have been studies done. I know that these studies came from here, actually WA, that they basically showed JAK-STAT pathways were differentially modulated when with the different subtypes. So I think that's an indication that certain intracellular events and signaling events are occurring. Does that answer your question?
Yeah. Other people's names I can get wrong. It really looks like there's an innate component to this, you know, from the work that you've got, and even from your early pictures, the tumours were much smaller at day 3 than.
Yeah, I mean, even in the rag mice, we think the innate system is playing a role with 2 and 9, which is focusing on the fact that we can get that elimination event. I want to put them all into the rag gamma chain knockouts. It's just time, resources.
So Chris Benedict from La Jolla published a paper on CMV a few years ago showing that the type 1 interferons really set the set point for CMV load in animals really during the first 36 hours of infection. So you may be seeing something very similar, but with tumor instead.
I mean, they're just remarkable compounds. Do you know what I mean? They have such biological, unprecedented biological activity. That's what got us started on here. Because I guess I was coming from the angle of antiviral immunity, and again, like you're saying, one of the first, sort of alarm bells that goes off is the secretion of type 1 interferon that gets the whole adaptive response kind of ramped up. And I've done a lot of work in the herpes system where, the immune system is very good at getting rid of the acute, viral load. So that's what got us sort of going down this pathway originally. So what we have done is we've just finished, we're just starting to characterize an inducible model. Okay. So what happens, I've shown you the results today mainly with nascent disease or occult disease. What happens when we have a palpable mass and then turn it on? Can we still see the same sort of modulation? I think that'll be a very interesting system, and it's sort of an easier system to work in. If you have a tumor and it starts regressing, can you then start doing flow cytometry experiments?
Do you have a feel for why the tumours don't seem to shut off interferon, but they do shut off tumour antigens?
Yeah, no, I don't know. Nope, not at all. Different mechanisms at play. I guess some of these systems, he's done it early on. He did a lot of explants, and you could still see a lot of GFP expression. At least I know with the 456 where we could get those tumours. I don't know.
The fact that you don't get cross protection from the tumour on the contralateral flank suggests that you need the continuous.
Yeah.
So you'd imagine that the suppression pressure is the same that you get from a direct antigen expression. So.
Yeah, look, that experiment was not a surprise, but it's the way it went, and it's pretty clear that whatever's happening in that microenvironment, hence why, as I was talking about, we're very interested in developing a cellular therapy that can deliver it to the tumour bed, see if we can get this effect.
There's no more questions?
Yeah. Again, about this about this local effect, are these molecules really secreted or presented?
Secreted.
Okay, so.
Yeah. So the assay that we do to sort of the characterization of the cell line was you take the supernatant and you expose the cell line to that titrations of that supernatant prior to infecting it with the virus. So can you see that interferon, as it was first sort of described, sort of gave uninfected cells the ability to stay uninfected. It interfered with the virus being able to enter and propagate. So that's sort of, we're looking at the secreted component.
But still surprising that on the counter flank, you see no effect. Because it could affect, well, it could expect that this interferon alpha is going to be secreted in the circulation.
Sure.
An effect spreading throughout the organism, not only locally.
Yeah, look, I mean, our first crude attempt at a cellular therapy was just to make our activated T cells secreted everywhere, right? So we just made them express it as the therapy and put them in, but we saw absolutely no effect. So I guess what we were thinking there is, alarm bells just going off systemically, and there's some really elegant work out there that shows you can actually transduce the liver in vivo, sort of, if you like, as an overexpression model, and you get large interferon secretion throughout the whole system, a whole lot of toxicity. And we know that with the interferon treatment that patients receive. It's very, very toxic. Some people say, I'd rather just die of my cancer than be on this stuff. I hate it. Whereas, the local effect could be quite dramatic, as you can see from these results. So we're definitely very different mechanisms at play. And it might even be in the viral infections, like herpes dividing, for example, locally in the skin, there might be a whole bunch of type 1 interferon that really promotes the immune system. Hence why we're going to start looking at the checkpoint inhibitors, effector function.
Okay, thank you very much.